for the entire immunology lot. I'm obviously recording this in a different style than the others, but I'm sure you'll soon get used to that. This chunk is all about the reproduction of viruses. You might in your GCSE days have heard of the lysogenic and the lytic cycles, and antibiotics and their overuse. So it's a bit of virus and a bit of what do we do against bacterial infections. So let's go. Specification reference here in case you want to screenshot. Main plan is the virus life cycles. It would have been a reading task, but I'll do it over video now. Regurgitating a little bit of something about the bacteria that you should know from the microbiology chunk. If we have time, we should be doing pool plates again and testing different antibiotic discs. So it would have been a nice practical. I'm hoping we're going to have time for that. Literally recap of aseptic technique, working by the Bunsen burner, your 10-centimeter radius, not speaking, and washed hands, washing everything down, sterilizing every loop in the flame, flaming the neck, all that, remembering the aseptic technique when doing microbiology. Now, we can test different antibiotic discs and how they will affect the growth of E. coli, for example. Hoping we will have time for that. Right. If we're thinking about viruses, what are they? Sometimes they're called the ultimate parasite because technically a virus isn't even alive. It doesn't fulfill all the movement, respiration, sensitive to change, growth and reproduction, excretion, nutrition, all the typical life processes. It doesn't fulfill those, particularly the reproduction part. It cannot do it by itself. It must hijack and use cell machinery. It's mainly the ribosomes that it needs. Sometimes it goes into the nucleus as well. Depends on whether you've got a DNA or an RNA virus. But key idea here is that we need the ribosome to make the new protein coating for the new virus, and we need to reproduce the DNA itself. So that is with regards to why it's not classed as living. So the reproduction part, but also the respiration. There is no use of energy forms like ATP. So no energy carriers as such, and all other respiratory pathways use that. How can a virus then reproduce? So by hijacking a cell or by using the cell's machinery and doing it in two different ways, the lytic and the lysogenic way. Let's do the lytic first. Key idea, the virus is adding its own genetic material into the cell. The genetic material of the virus is then being read and translated at the ribosome in order to produce the new viral material. Now, the lytic cycle is when the host cell actually bursts. I'll do that one first, when it's actually breaking open. You have learned about electrolysis, hemolysis. Whenever you have lysis, lytic is breaking. So if you look at this diagram here, that shows the lytic cycle. So the virus somehow finds a receptor where it's got a complementary shape to it. It will find a way of getting its nuclear material into the host cell. If it's just RNA, then it's fine to just have that in the cytoplasm directly. It may be a DNA virus, and that might have to go via the nucleus. So different story there. The ribosomes are doing their thing and producing the new protein codes and so on. Let's ignore how the DNA, how the RNA is being doubled up. Let's ignore reverse transcriptases, all making things complicated. Let's just pretend we are getting new virus particles, are being assembled, and in order to release those virus particles, the cell actually bursts open, the membrane bursts open, which means afterwards that cell is no longer living. It's literally spilling all its contents to the outside, including all the viral material, which means you've literally got a gap between all other functioning cells at that point there. The other option, that would be the budding option, what you've got in this diagram here. If a virus is assembling the DNA in its protein coating right by the cell membrane, it can use a bit of stealth tactics. It can wrap itself up in the cell membrane of the host cell, which is a beautiful way to avoid the immune system catching you if you've actually got literally a cloak of invisibility on to the immune system, because the proteins presented to the outer surface at that point are very, very similar to the actual cell that was invaded in the first place. This is we're going to speak about influenza a bit more anyway, when you've got the hemagglutinin, the neuroamidase. You might remember the wording of spike protein every time you're speaking of COVID-19. So that's on this diagram as well here. The lysogenic cycle, instead of the lytic cycle we're breaking out. The lysogenic cycle, that's where it becomes a little bit more sneaky and long term, a bit even more stealth mode. If the DNA from the virus is actually being incorporated into the genome of the host, so into the actual DNA. For us, I believe it was 8% human DNA is thought to be of viral origin. It can then stay there for many years undetected and just not being copied, not being referred to, nothing's happening to it. It may be that a secondary infections, in the case of chickenpox, that is what's happening. And you actually hold on to a piece of the chickenpox virus in your DNA, only to then later on, if the shingles virus is somewhere around, then a secondary release is happening. So there are viruses that do it a bit more complicated. If you get a weird scenario, go with it, go with the scenario that they're giving you. So that lytic cycle, yes, it is about 8% of human DNA that is thought to be a viral origin. So obviously made it into the stem cells, not stem cells, into the, oh, I've lost the word, not the somatic cells, the germline. That's what I was looking for, into the germline cells. Right, this one here, I actually wanted to get you to do this as a quick reading task in class, but as it's lazy learning and you're doing video, if you want to do it properly, please pause the video and identify in what different ways, 4 different ways, wouldn't that be an ideal 3-marker question with 1 to spare, 4 different ways in which viruses actually cause you to be ill. Pathogenicity is actually how do they cause illness or disease, rather than just having a virus and a virus does nothing. This is about how does it cause disease. So cell lysis, toxins, transformations, and the immune suppressants. So I'll go through them with a couple of examples slowly. If you wanted to take a screenshot, I think you've had enough time by now. The first one that does cell lysis is a very typical one. You may have heard the word rhinovirus for the typical common cold. There are 200 different versions of that running around. And it literally causes the cells in your upper respiratory tract, that's your sinuses, your throat, your nose, and maybe part of your trachea and bronchi, to just have cells breaking, bursting open, releasing more virus. The idea of that virus is obviously that you're exhaling the virus particles again, so that somebody else can have the joy and pleasure of those. So cell lysis, rhinovirus, and just breaking and leaving gaps in your respiratory tract. Hurts, will need to be replaced. You've got inflammation, and. The next way in which you can have pathogenic effect from a virus is that they're releasing toxins. You may want to research Botox if you, just on the side, Botox and the injections of Botox to numb nerves, both for a medicinal use as for beauty surgery, the injections in the face to stop wrinkles from appearing. But the ones you need to know, according to your syllabus, you've got measles, where the toxin itself can actually cause chromosomes to stick together and fuse, usually in the somatic cells, so that is usually not something that affects a germline. However, it causes cells to no longer do what they're supposed to do, no longer replicate, no longer being read. Difficult situation, but hey, here we go. Herpes, that one causes cells themselves to fuse to one another. And the last one is that you've got toxins that somehow interfere with the cell machinery. So they're attaching themselves to RNA, rendering messenger RNA or ribosomal RNA, rendering that useless, inhibiting DNA, stopping the whole protein synthesis apparatus, attaching themselves somewhere to a ribosome and making that stop working. Third out of the 4 is cell transformations. You have met TP53 before when we discussed the cell cycle and the checkpoints in the cell cycle, when you've got mitosis and do we now move on to the S phase? Do we move on to G2 and so on? So you need to have those checkpoints. Now, the tumor suppressor gene is obviously making a protein that does that proofreading role. If you insert virus DNA exactly at the place where that tumor suppressor gene is, and the human papillomavirus does that. It then can cause cervical cancers. Human papillomavirus, that should click together. You ladies were offered a vaccine in year 8 against HPV, human papillomavirus. That is that one. And the last one out of the 4, you have got the immune suppression activity. So a virus can slow down how your lymphocytes mature. That means slowing down how antibodies get produced. The second thing they can do is, particularly HIV, the virus that then will later on cause AIDS. HIV destroys T helper cells. T helper cells are involved in helping B lymphocytes to produce antibodies, all kinds of antibodies, not just against HIV, if there was such a thing, but lymphocytes will generally produce antibodies against common colds, against other things. Sadly, AIDS sufferers, where actually HIV then gradually turns into the actual illness AIDS. The common causes of death are either cancers or tuberculosis or common colds, things that usually your immune system has a bit of a check on at least. Many cancers are being detected by your immune system in early stages, dealt with, gotten rid of. It's only the ones that your immune system doesn't catch that will turn out into full-grown tumors. The last thing they could do, it could stop phagocytes, so your neutrophils and macrophages generally, to do that flowing around a phagocyte, so that phagocytosis, that engulfing, is reduced. Right, I'm going to stop here to do the antibiotics in the next video.